In the human body, there are groups of cells called mitotic cells, ones that require cell division to repair and regenerate tissue. There's another group of cells called post-mitotic cells, ones that do not undergo mitosis. These mitotic cells, such as endothelial cells or fibroblasts, undergo mitosis to pass on the genetic material. This requires DNA replication, and in every mitotic cell except stem cells, telomeres are lost during DNA replication. A single DNA sequence, TTA-GGG, that is repeated hundreds of times on the ends of chromosomes, are called telomeres. They are mostly found in eukaryotic species and do not code for any protein. They are simply there to protect the DNA from any damage during the replication process. DNA can become damaged because during the replication process, the DNA polymerase cannot copy the very end of the lagging strand. This happens when the replication fork reaches the end of the chromosome and there is a short strand of DNA left. The RNA primer cannot get the Okazaki fragment started because the primer would just fall beyond the chromosome end, and hence the section of DNA strand cannot be replicated. This can lead to damage, thus increasing further complications. The ends of the DNA strand not being replicated results in chromosomes shortening each round of replication. This could cause a loss in important genetic information if critical genes are not replicated, and hence telomeres are present to prevent the damage of DNA. In 1961, Leonid Hayflick proved that cells can only divide a finite number of times. This is known as the Hayflick limit. Cells can divide 40 to 60 times until telomeres become too short. After that, the coding DNA is at risk from further replication. Our body has a contingency plan built in which the cell initiates a DNA damage response. This means that it gives up its ability to divide, becoming a senescent cell. Thus, cells transform to prevent impending DNA damage that can lead to cancerous cells or tumors. However, as people get older, tissues can't repair themselves as well because of the lack of cells that perform mitosis. If all the cells stop dividing, then wouldn't this have a negative impact on the functions of important organs and tissues? Well, some cells can actually reverse telomere shortening by using the enzyme telomerase, which extends the telomeres of the chromosome. Some examples include stem cells and cancer cells. In telomerases, the RNA template contains the sequence CCCUAA, the complementary base pairs of TTA-GGG DNA that forms the telomere. The telomerase allows the cell to make DNA copies of this RNA template. Essentially, the 6 nucleotide DNA repeats can be added to the chromosome end, lengthening the telomeres and allowing the cells to divide for a longer period of time than what was suggested by Hayflick's limit. These enzymes are usually not present in somatic cells, but are active in cells that create sperms and eggs. This is why stem cells can divide constantly, which creates an entire organism. Studies show that if telomerase is inhibited by drugs used in cancer therapy, the excess division of cancerous cells could potentially be stopped. Okay, but you might be wondering, how did telomeres and telomerase even get discovered?
In 1513, the famous explorer Jean-Ponce de Leon believed he discovered the Fountain of Youth, a magical water source that supposedly could reverse the aging process and cure sickness while searching for Florida. Although the Fountain of Youth is a myth, the discovery of telomeres can be used to explain aging. Centuries later, in 1912, Alexis Carrel, a surgeon, established a culture of chicken heart fibroblast cells, which he grew in a laboratory for 35 years. He concluded that vertebrate cells can divide infinitely in culture. As individual cells, they are immortal, and that aging is an attribute to a multicellular organism as a whole. In 1961, the concept of immortal cells was challenged by Hayflick and Moorhead. They discovered that fibroblast cultures from a human skin could divide 40 to 50 times and then undergo senescence, which simply means that the inability to divide any further. Further work also showed that cells in the elderly divide less than younger people, which suggests that Alexis Carrel's theory was based on the total number of divisions since birth and not the total divisions in culture. Questions emerged after this discovery about the relationship between the limited capacity of the cells to divide and aging, or if the mechanism that stops the cells from dividing is to prevent tumor formation. In 1972, James Watson recognized that the end-to-end joining of DNA in a bacteriophage allows the complete replication of the bacteriophage genome. Linear phage genomes could avoid the end replication problem by joining multiple genomes before replication and reducing the number of actual ends, hence minimizing the damage by incomplete replication. At the same time, Alexei Alognikov was also thinking about the same idea, however believed that the problem was at the beginning of DNA replication, and Watson thought it was at the end. In 1978, Elizabeth Blackburn and Joseph Gall were observing protozoan tetrahymia thermophilia, which creates around 20,000 telomeres while undergoing meiosis, whereas human somatic cells only have 92 telomeres at the ends of the chromosomes. They discovered the enzyme that extends telomeres and hence naming it telomerase. Blackburn removed the telomerase and noticed that the telomeres get shorter after every cell division, eventually leading to the organism's death. In present day, the knowledge and research of telomeres and telomerase has progressed far enough that there are many economical and societal benefits.
For example, when bone marrow is transplanted, the donor cell age much faster than they would have if it was still in the donor's body. The more cells that are transplanted, the less the telomeres shrink. This suggests that transplanting only a few cells stresses them in their effort to repopulate the recipient's marrow, and in response, the cells age much faster than normal. This might explain the increased risk in the patients being diagnosed with leukemia years later of their bone marrow transplant. Research on telomeres and telomerase would prevent this since the bone marrow would be able to replicate without aging as fast. Telomerase might be used to treat AIDS and HIV. As the HIV infection progresses in a patient's body, the T cell count also reduces. This makes it hard for the patient to fight HIV. To prevent this, hematopoietic or blood-forming stem cells could be taken from the patient at the early stage of the infection. The cells would have their telomeres extended in culture, and then when the patient's T cell count reduces, they can receive the stored stem cells that can replenish the T cell supply. This can potentially save numerous lives.
Along with benefits, there are also negative side effects. Telomerase is proposed for gene therapy to extend the lifespan of cells in vivo. However, cellular senescence could be a mechanism for tumor suppression. Thus, allowing the cells to further divide could potentially increase the risk of tumor. Adding telomerase to increase the telomere length can possibly activate or deactivate an oncogene based on what is in the way of the telomerase.